All right, so welcome to the day 2, lecture number 4. So we are going to talk about emerging technologies that can be used, those can be used to extract proteins from different biomasses. In the previous lectures, we discussed about conventional methods, salt extraction, dye-fractionation, and so on. Now we talk a little bit more about the novel technologies or emerging technologies, okay? Some technologies I use novel because not well established. Sometimes they also use the word emerging technologies because again the reason is not so well established, especially in an industrial point of view. However, that is being done, I mean the work is done in the R&D level, research and development level. For example, a lot of laboratories use some of these some of these technologies to extract proteins. Okay, I'll try to explain which technologies are actually scalable, which are not, what are the advantages and what are the disadvantages. Again, I start with the basic principles of their operation, then I'll show you some fundamental examples, some case studies and so on. Then again we discuss about advantages and disadvantages. Is it okay? If there is any question, raise your hand and ask. All right, let's begin with the ultrasound assisted extraction. I'm pretty sure every one of you have heard about this one, right? It's one of the most well investigated non-conventional extraction methods today. I can say that one. So basically ultrasound is actually a sound wave, right? You know about sound. When I speak, it's a sound. Every wave has a wave properties. You know that it has wave number, frequencies, all these things you have studied in physics, right? Ultrasound is a special kind of sound wave. The difference is that basically the frequency is like 2 to 10 megahertz and less than 100 kilohertz. Basically, this is the high frequency ultrasound is basically used in diagnostic purpose. If you go to clinics, they take ultrasound, they do some testing to see where the baby is moving and so on. And perhaps there they use this kind of frequencies. But in food science, for the extraction purposes, we use low frequency, high intensity ultrasound. Basically, we are talking about the intensity in the range of 10 to 1000 watt per centimeter square. The frequency is low, but the intensity is very high, and that can damage the tissues and extract the proteins out of the tissues because of their high intensity. Okay? And remember my previous explanation, we have a cell, we have proteins inside the cell. We need to bring protein from inside to outside. What do we need to damage here? Cell wall we need to damage. Okay, it could be plant cell, it could be microalgal cell. The cell wall has to be damaged. So conventionally we are using different technique. Non-conventionally here we are using ultrasound because ultrasound apparently can break the cell wall. Let's see how exactly that does. Okay. So two types of ultrasound equipments are available today. One is the bath type, which is almost all chemical laboratories will have this bath ultrasounds. We call it as ultrasound cleaners basically. It's used to clean the system because it basically, because of the sound waves, it will reduce the particle size. For example, if you want to solubilize protein, if you want to solubilize polyphenol, if you want to solubilize amino acids, then you take it in a tube, put it in the ultrasonic bath and turn it on for 5-10 minutes, it will rapidly solubilize. Why? Because basically it reduces the particle size and improves the solvent solute interaction, right? Then they improve the solubility, and improve the solubility means actually increase the yield. Do you agree? Solubility is nothing but yield in our case. Increase and more solubility and we get more and more yield. Then there is something bigger ones called probe type systems. Basically these are more powerful and efficient than the bath type. In the bath type what happens, the ultrasound comes from the wall or from the bottom. Okay, sound waves first comes to the solution which is present outside the bath, basically it's water, right? The ultrasound first hits the water and then it comes to your matrix, right? Basically you already weaken the power here, right? But in case of the probe type, they take the, let's say, solution here, the matrix and the solvent and the solute, and they insert the probe directly inside the sample matrix. And then the probe, the transducer gives the sound waves and that is directly interact with the solute and the solvent present in the sample. So it is more powerful and more efficient. If you really want to do the extraction studies, we recommend that you go for the probe type. I don't say you should not use bath type, you're welcome to use, but it's less efficient compared to probe type. And bath is extremely cheap compared to probe. This is a specialized equipment, they are more expensive compared to bath type. Depending on availability in your lab, you use whatever you want, but for the large scale protein production, we often use the probe type. Is it clear? All right, so now I'm going to explain you how exactly the extraction takes place, okay? Remember, I have a beaker and I have a solvent, right? And my cells are here. These are my plant cells or tissues, matrix, microalgae, I don't care, right? Inside that I have protein, right? Inside that I have protein. Now I put an ultrasonic probe inside this one. This is the probe that generates the ultrasound waves. When it generates the ultrasound waves, the wave propagates in this way. We all know that the wave, how it propagates, right? When ultrasound wave propagates inside the solution, they create a negative pressure and the positive pressure, right? Positive pressure region is called as compression. What exactly here happens is that during the compression type, the solvent molecule that interact much readily, there is a more interaction between solvent molecule, and in the rare fraction cycle, they just try to go apart, okay? Together, apart, together, apart. That's what continuously happening. Together, apart, together, apart. At some times, the negative pressure increases, then they go apart, and basically at that time, they create a small air bubble, a vacuum, the vapor filled bubble. Okay, inside the solution there will be formation of very micro bubble. You can't see it, it's not a big one this size. A micro bubble will form. If we are continuously applying the ultrasound, basically the bubble will slowly increase their size, okay, and the vapor is filled inside the bubble, okay, because you're continuously having this rare fraction cycle, so but the bubble will size increase. At a particular time point, a particular size when it reaches, it cannot increase again, so then you have a compression cycle, the bubble will implode. It's not explosion, it's an implosion. The bubble will collapse, right? When the bubble collapses, it releases energy in the form of heat. And they say that the temperature can reach all the way to 5000 Kelvin, otherwise 5000 degrees Celsius, extremely high temperature, but in a very localized region. And the pressure can be up to 50 megapascals, it's also high pressure. Now you create a high pressure and temperature right next to the probe and here, right? Now you have a cell here and you have a high temperature and high pressure, the small regions that is created. Now because of that temperature and pressure, the bubble, what we call microjets, very small microjets are created, and that will damage the cell. That will explode the cell, releasing the components from inside to outside. Does it make sense? This is the basic principle of ultrasound. This is called cavitation. The process is called cavitation, okay? Basically the waves propagates and they produce micro bubbles filled with vapors. At 30 particular time point, they cannot grow further, they implode. Implosion will create microjets containing high temperature and pressure, and that will damage the tissue or cells, releasing the intracellular compound into outside, and that is what we call extraction. So ultrasound is nothing but an extraction assistance. It's not a real extraction, I would say. It assists the extraction by breaking down the cell walls or plasma membrane, whatever it is. Clear? Any question? It's the most simplest way I can explain this ultrasound. I can go deeper into subject, but that's not needed, okay? Here is the microscopic image. This is a control sample and this is a microalgae. So you can see that how it is actually damaging the cell. Once the cell damages, things comes out, that's called extraction. Coming to the equipment, so it's very simple. This is the bath type ultrasound here. Bath type operates around 40 to 45 kilohertz, and basically there is an ultrasound transducer, they produce sound waves, ultrasound waves here, and this is where water, and you put your beaker containing your protein matrix, you can do extraction, right. And the probe is very simple, you have a probe made up of usually stainless steel and produces ultrasound waves and damages the cell. Usually ultrasound is used in the batch system. Batch system means you have a solution, you treat the system, remove it, finished. That's often it is used, but there are some studies they show that you can also make a continuous system, means you continuously supply the matrix, what we call feed here, do the extraction and remove the feed. You can do that one, but I'm not very sure how feasible it is. It has its own problems. Okay, the batch type is most often used, but for food industries, tell me which one is more efficient? Continuous, because we need to save time and money. So this is something we really need, but this is we are at the moment. Okay. So this is a simple curve that shows how the extraction yield would look like. In ultrasounds, usually the yield increases with time, the time with ultrasound treatment time here, and then the yield can decrease. And sometimes the yield can continuously increase, then can come to a plateau. Now can you tell me why the yield increases at some point, until some point? Increasing ultrasound treatment time increases the yield, why? More explosion happens, continuously explosion action, any other components will increase, because you have more and more explosion, more and more cell damage, more and more things are released. And why is there is sometimes there is a decrease in the yield? For example, this is polyphenol, okay? Why the yield is decreasing? Please note that people call ultrasound is a non-thermal technique, but if you continuously apply bath, you just see that the bath, you put your finger, right? It becomes warm and hot, right? Usually some biomolecules are highly sensitive to temperature and they can be damaged if you do a continuous ultrasound treatment. You increase the yield, yes, but they're also damaging them, because extraction and degradation goes in parallel. In this case, extraction rate is higher than the degradation rate. In this case, extraction rate is lower than the degradation rate. So degradation is overwhelming here. That's the reason why you see the loss. But proteins is usually not a problem, because proteins are stable. Okay, but if you're using ultrasounds for polyphenols, anthocyanins, bioactive compounds, you have to play around with the parameters. Okay, usually this is the case for proteins. Continuously increase, then nothing to extract any longer, then it stops. All right, so this is one of the experiments we did back in the days with Chlorella vulgaris. Chlorella vulgaris is the microalgae that produces proteins. We try to extract proteins from Chlorella vulgaris. It's one of the most cited paper. We did a very simple work actually. What did we do is that we tried to compare whether or not ultrasound is beneficial to extract proteins. We did the conventional extraction with the different I think alkali and acid, and then we did the sodium hydroxide, then we did the ultrasound assisted extraction. Yield was very similar, can you see? Because the maximum you can extract is only 70%, no matter what. But the only difference we saw actually the time. Basically, you need to wait until 6 hours or 24 hours, but you can do it in 5-10 minutes. I don't say that ultrasound will increase the yield, but basically it becomes process more efficient, right? Basically, time is money in industry. Time is money, right? So within short time, you can get more yield. It's not necessary always that ultrasound will increase the yield, but it is an aided technology. That means it helps, so you can reduce the time. It's a good take-home message for you, right? If you want to do it quickly, because for example, it's just simple, the same process, like you want to dissolve some amino acids, you can shake it for half an hour and it still dissolves after half an hour. But if you put an ultrasonic bath, 30 seconds it dissolves. That's where you save the time. That's the advantage of ultrasound. And there are a lot of parameters you can control for in the ultrasound. I don't go in detail at the interest of time, because you have a parameter to control the frequency, we have the parameter to control the intensity, and we have a parameter to control the time, and with a parameter to control the pulses. A lot of parameters you can control, but usually the intensity is the governing factor and the time, right? And if you apply more and more power, you can get more and more yield, and or less time you can, within less time you can extract proteins or biomolecules. Is it clear? So a lot of parameters to cover: power, intensity, time of treatment, solid to solvent ratio, many things you can control. This is what people often do. There's thousands of literature on ultrasound on these parameters. A lot of response surface diagrams. Have you seen that one? They talk about, oh, how it is increasing. I did not write all these things because I thought it's very boring, because you can find it in the internet. So what your take home message is, you can control any parameters. However, usually the power and the time is the most governing one. The solvent and solvent ratio and all you can change to improve the extraction yield. Okay. Here I tried to summarize from this one. You can see that many plants sources have been used. Ultrasound treatment has been done on the many plant sources to improve the yield. You can see that people talk about 37% increase, 13% increase, 10% increase, so much percentage increase. It increases. Okay, it's simply because it helps the damaging of the cell wall. It will help the reduction of the particle size. Please note that particle size is very important, because if you have a larger particle size and you have a solvent, and if you have a smaller particle size and this is a larger particle size and a smaller particle size, which one dissolves efficiently? Smaller, right? And ultrasound will help to reduce the particle size. That means there is a more interaction between particle and the solvent, because extraction is nothing but interaction between the particle and the solvent. So that interaction is enhanced because of ultrasound, so you get the better yield in addition to damage of the cell wall. Okay. All right, so can you mention one or two advantages and disadvantages? Ultrasound, what is the advantage? Yeah. The time is money, it's less time consuming, the process efficiency will be higher. I agree with you. Any other advantage? One other claim is that ultrasound is a non-thermal process. Yes, ultrasound is a non-thermal process provided you maintain the power and temperature, power and time properly. Otherwise, usually the heating effect happens. I showed you that 5000 degrees Celsius. If you continuously heat, solution start boiling also, like it's super difficult. But ultrasound can also, you can also actually have a water chiller. Basically, you can control the temperature. So then you can prevent the degradation of the molecule. So the technology itself is non-thermal, provided you control the treatment conditions.
There is disadvantage that installation cost is high.
The capital installment, the
yes,
capital is
cost.
very high. Baths are not that high, it's cheap, but the probe systems are actually a little bit expensive depending on the size and the model. So I'm investment. Agree with you. Temple size is also limited. You often used in the batch system. If you really want to have a continuous system, you need to redesign it completely depending on the need. You don't know what is the flow rate and solid to solvent ratio. Everything has to be optimized. It's not a universal method here, so it's not an easy one. I can tell you that ultrasound system is used in many cases, but it's not industrially well adapted yet. Okay, because some part of the world is still comes under the novel food category. So especially in the EU, so it's still some industries are using, I know, but it's not that every single protein industry is using ultrasounds. That's not true. Okay, and another disadvantage is that the ultrasound probe what they use, apparently they say that after a long-term usage, if you're using it in acid solution and a base solution, the stainless steel or the steel that start leaching the metal ions and so on, that might have some problem. And not only that, the probe what you use here in the solvent, and you have water molecule here, right? And the temperature I said 5000 and pressure some 50 megapascal here. And basically the water molecule is H2O, is basically HOH, right? It can have a homolytic cleavage because of ultrasound, and you get OH radical plus hydrogen atom. And OH radical is extremely notorious radical that can oxidize every single molecule, right? Super reactive, it does not discriminate protein, lipid, polyphenol, just attacks whoever comes right in front of it. That's one of the challenges, oxidation, lipid oxidation, protein oxidation, polyphenol oxidation can happen because of this one in extreme scenarios. And this is a strong reducing agent. This is strong oxidizing agent. So there is some chemistry problem as well. Okay, is it clear? Ultrasound is a good technology, increases the yield, but use it carefully. Can be used in the batch, but industrially it's not yet well established, still under the developmental stage. Then I will move on to the next technique called pulsed electric field technology. Have you heard about pulsed electric field technology? All right, it's not similar to ultrasound, but it's an equivalent technique, I would say. In ultrasound we use sound waves, right? In pulsed electric field we use electric field. That's the only difference, right? What we do here is that we take a sample. It could be your microalgae, it could be plant extract, it could be potato, it could be carrot, anything can be placed in principle here in a solution. It will be water. There should be a solution is required, and that is placed between two electrodes. Okay, and we apply very high electric field. We are talking about up to 20 kilovolt per centimeter. It's actually extremely high field strength, right? And that pulse is given, it's not continuous applying the voltage, it's basically pulse. They give an electric shock basically. And we're talking about in nanosecond to millisecond, and the number of pulse can vary between 10 to 1000 or something depending on the type. So basically they're giving the electric shock continuously like this one. And that apparently damages the cell. Am I clear? So they are giving high electric pulses for a very short duration for a continuous time. Okay, it's not a continuous one, but a short pulses they give. At that time, what happens? That the principle that electroporation happens. Electroporation. Poration means formation of pore. Electro means electricity. Basically, the cells will have some pores because of this one. Now, protein is inside. When you have pores, what happens? They leach out. That's it. It's again an aided technology. It doesn't damage completely like ultrasound. It creates small pores on the cells, and apparently that helps to extract biomolecules, including proteins, so if the pore size is sufficiently big enough. All right. Now, how exactly that pore happens, I'll take it only 30 seconds for that to explain you. You put the sample, the biological cells in between the electrodes, right? These are the two electrodes, right? When you apply the positive and negative voltage here, basically there is a formation of electrical charge, the layers. Inside the cells there will be lot of ions, right? Some are positively charged, some are negatively charged. And outside you are applying the voltage, they basically start organizing as a dielectric barrier membrane now here, okay? And there is a particular electric voltage they maintain here. If you continuously apply this electric field at a particular electric field strength, the transmembrane potential, that is what the potential generated over here, is exceeding, and that will lead to the damage of the cell membrane. I'm not talking about the cell wall here, it's a cell membrane, basically plasma membrane. So there is a positive charge here and the negative charge here, and negative charge here and the positive charge. They have the transmembrane potential. And when you applied an external electric field strength over the transmembrane potential, they rupture. And the rupture is actually gives the pore, the hole. Sometimes that's called electroporation. The hole can be permanent, that means reversible, and it could also be irreversible depending on what the field strength you are applying. If it is reversible, basically the cells can rejuvenate. You can actually regrow that one. If it's irreversible, their cells will die. Basically they leach all the cytoplasmic material outside, including the proteins. Okay? And the electroporation is the basic principle here. Ultrasound cavitation is the basic principle. Here the electroporation. This technology is also used in cell and molecular biology, you know. In molecular biology, they want to put a DNA from outside to inside. We studied molecular biology, biochemistry. They can use the same technique here. But we want the inside to outside here. That's the only difference. The protein, we want to get it out. I have some illustration to show all these things. I don't know, I have already explained this one, how the membrane opens up here and the things from inside to goes outside. Okay. This is some of the examples, microscopic image, just to show that how chlorella vulgaris cells will be damaged. They, I think in the paper they show that there is a hole formation here. I don't believe it exactly, it's really that hole because it looks so beautiful here. But basically the damage can be seen by this these cells here. Basically, when there is a pore formation takes place, they lose the cytoplasmic material, they lose the osmotic strength, osmotic balance, then they shrink. The shrinkage of the cell is direct indication of cell damage, okay, and that shows extraction of biomolecules. Is it clear? Shelf will shrink because of loss in the osmotic pressure. These are the commercial equipment, how it looks like. This is, I took it from the internet, but we have an equipment in our laboratory, something similar to this one. Basically, it's a big machine because you have to do a lot of electrical stuff here because you're talking about 20 kilovolt per centimeter. It's a lot of electric field strength. And actually the cell is inside. Basically, it's a small piece of Teflon cell with the electrodes inside, and you put a sample. You can put carrot, potato, anything you put in solution, you can actually do. Historically, it's not used for the extraction, just to let you know. It's used for the drying purpose, because you damage the cell and you make a hole, and the drying becomes much faster because the water molecule from the cells can evaporate much faster. So it's used for the potato a lot of drying technology, and also to soften the food material, because when the tissue is damaged, it becomes more soft, then it has a different functional property. But people are also using it for the extraction. So you can see that the damage increases with increasing the electric field strength. Just to remember, just to show you that one of the important operating parameter is field strength. Increasing the field strength will increase the damage up to a particular degree, and then no longer you can damage the cell. Yeah, just to show here about the efficiency of pulse electric field. I tell you that PEF or pulse electric field technology is used in the, it's not, again, not industrially well established. It's still in the research and development phase for the extraction purpose. It's industrially established for the drying purpose. It's already used in many industries, but not for the extraction purpose, to the best of my knowledge. So when compared to high pressure homogenization, you know the technique they use, high pressure, the PANDA, the high pressure homogenization is a normal technique used to break down the cells. It's not that efficient. So it can be used, but PEF is not a really efficient technique, especially to extract proteins from cells that has very strong cell wall. Please note that cell wall cannot be damaged by PEF. Cell membranes can be damaged by PEF. To damage the cell wall, you have to apply extremely high electric voltage, and that we call high pressure, high voltage discharge. It's another technique, but it's not very energy efficient. Am I clear? To extract this molecule from inside to outside, it's not sufficient if you break the plasma membrane. You need to break the cell wall. But usually PEF cannot damage the cell wall efficiently. So it's not a really an efficient technique to extract protein, in my opinion, according to the literature as well. It does some improvement, provided you already damage the cell partially, then you can do it more damage with the PEF. It can be used as an aided technique. Okay. This is an experiment showing that how the field strength increases the yield. You apply more and more field, you get more and more damage to the cell, and more yield can be obtained. Okay. Okay, what are the advantages and disadvantages of this PEF technique according to you? I can conclude, I can summarize. PEF is nothing but it works based on the electroporation of the cell membrane. It makes the hole on the membrane because of the change in the transmembrane potential, and through that hole the proteins can come out. Okay. Then the extraction yield can increase. I don't say that it is a fantastic technique to get the highest yield in the world. I don't claim that one. It's not so efficient with the microalgae. We also tested it. It's very inefficient with seeds, for example, because seeds have such a strong seed coat. It's very difficult to make a hole on the seed coat with PEF. So it's not so efficient. We tried it with rapeseed. It did not really work out. So, but if you partially damage it with milling and other things, you can still increase the yield. Okay. What is the disadvantage and advantage apart from this one? Samples needs to be in solution. You can't have it in a dry state. That's very important. Yes, anything else?
Cost.
Cost, the PEF equipment is really expensive. It's a capital investment, and then it can be done in batch and continuous. That's a possibility with PEF. We can do it both batch and continuous with PEF. The equipment are made for that one. The other disadvantage here is that the pore sizes actually matters. Basically, if you look into the, let's say, an animal cell here, it's easy to damage animal cell with PEF. Um, the proteins are actually big molecules. Do you agree? And polyphenols are very small molecule. Imagine, I mean, animals do not have polyphenols. I'm just giving an example. The small molecules can easily come through. For the proteins to come out of these pores, that have to be sufficiently wide enough, but that is not always the case. So you need to optimize it a lot here, and it's also energy intensive process. Is it clear? PEF. You're welcome to use PEF to extract proteins, but if you don't get a lot of yield, don't blame me. Okay, that's the conclusion. Then the last techniques for today is enzyme assisted extraction. It's again a very well investigated technique. Okay, there is 10,000 articles on enzyme assisted extraction, not only for proteins, polyphenols, anthocyanin, pectin, carbohydrates, peptides, minerals, whatnot. Everywhere they're using enzymes. Again, coming back to my very first slide, this is the cell and the protein is inside. Who is the barrier here? Cell wall. Everybody is trying to break down this cell wall, right? Now, I told you cell wall is made up of what in plants?
Cellulose.
Cellulose, okay, and pectin and hemicellulose, okay. Basically, they are very complex network that is protecting the cell, right? Now, if I use an enzyme that can hydrolyze, that can degrade, that can dissolve the cell wall, basically the cell will open up like this. Do you agree? Then the protein will immediately come outside. That's the whole idea of enzyme assisted extraction. Now tell me which enzyme you will use to hydrolyze cellulose? Cellulases. That's something you studied in biochemistry. Aces are enzymes, right? Hemicellulose is a carbohydrate. Which enzyme will you use to hydrolyze it?
Cellulase.
Hemicellulase. Pectin is a carbohydrate. Pectinase. Okay. And protein is also some for example microalgal cell also contains protein on their cell wall. Which one you will use? Proteases. Basically what we use in reality, we use combination of these enzymes, not only cellulase, not only pectinase, not only protease, we use the combination of the enzymes to hydrolyze the cell wall to bring down the protein out. That's the only the phenomenon here. Is it clear? A lot of enzymes are available commercially, and this technique is used commercially also because it's easy to upscale this one. You can do it in a big reactor, you can do it in a small reactor, you can do it in open reactor, you can do it in closed reactor. It's easy, and enzymes are commercially available. You just buy, add, and do the job. Okay, I go a little bit in detail how exactly it is done. You take the protein as a flour, mix it within your solution, and add enzyme and shake it. Things will happen, cell wall is damaged, you get the protein out. Again, it's an aided technology. You can use an sodium hydroxide here, provided enzymes do not die here. Many things you can use it in combination. Is it clear? It's just damaging the cell wall. I have a small, I don't go in detail for this one. This slide you can read it at home. It's not, but I just to show that for lignin, we have different enzymes. For cellulase, we have different enzymes. We have hemicellulose, we have different enzyme. We have pectin, we have different enzymes. For chitin, for example, insects have chitin, and also I think fungi also, the cell wall is made up of chitin. Then you have these enzymes. They're available in different combinations. You can buy it as a solution, you can buy it as an immobilized beads, depending on your application. Then you can buy them and you can use them. All right. Just to show one example is that it's not often a single enzyme doing the job. Just to take cellulose and an example, cellulase is a very complex polymer made up of glucose, but there's having a different network, all right? And initially you have endogluconase and cellobiol. This is, imagine this is a cellulose network, okay? Usually the endogluconase will actually cleave it here, and then you get an oligomers of cellulose like this one. Then there is a beta glucosidase, that is an enzyme that convert oligomer into monomers. then the hydrolysis happens. Same thing is true with hemicellulose and pectin. You can go through this slide later. Just to let you know that cellulase is not a single enzyme, it's a combination of different enzymes. They do different functions. They can cleave internally, they can cleave on the side part, they can cleave dimer into monomers. So you need to use the combination of enzyme to completely cleave into hydrolysis. But for extraction purpose, you don't have to do the complete cleavage. Do you agree? What is the problem if I completely cleave the cell wall? All things in compound, but if I, for example, if I convert every cellulose into glucose, what is the problem? Of course, everything, now the cell wall is completely dissolved, okay? What is the problem? Now I have a beaker, I have a cell, I have a protein, this is cellulose, okay? This is cellulose. I'm converting every cellulose into glucose. Now I have protein and lot of glucose here, right? Separation again will be a problem, exactly. Cellulose you can precipitate by centrifugation because of fiber, right? It can be centrifuged and precipitated off in most cases. But if you convert into glucose, it's completely water soluble, then your life is gone. Then you have to again use another technique to remove the glucose once. And not only that, the chemical reaction, glucose is highly reactive with protein, Maillard reaction. If you do it heating, glucose and protein react to undergo Maillard reaction, you get beautiful caramelized smell and other smell and everything. That's we don't want. So you need to control the processing parameter to get the thing what we really want. We just want it to open up, not completely dissolve. But if you're extracting glucose, probably you do this experiment, but that's not our aim. We want to extract proteins. This is the same mechanism, it's already in the slide, and this is a microscopic image showing the cell wall dissolution, that this is a cell wall with a staining and you don't see the cell wall after enzymatic treatment, okay? Okay, the enzyme kinetics I will not go in detail, but you all know that every enzyme has its own optimal conditions, right? They come with these bell-shaped curves. They have a pH optimal and they have a temperature optimal. It's recommended that you work in this particular region, otherwise enzyme will be denatured. Sometimes they are reversible denatured, sometimes they are irreversibly denatured, the enzyme will be damaged. So try to use the optimal condition. But it's always recommended that you don't work in this region. Can you tell me why? It's okay to work in this region in the enzyme, but not in this region. The denaturation, high temperature will irreversibly damage. If you really want to reuse your enzyme, then you can't use it, right? If you really want to here you will compromise the yield because enzyme is not super active, I agree, but enzymes are not dead here. If you want to reuse the enzyme for some other purposes, basically it's nice to use in near optimal condition, not on the right hand side, but on the left hand side. Make sense? So don't try to target this particular, this is the danger zone actually. You get the highest activity, but you're also killing the enzyme here, okay? And this is a substrate effect. Basically, if you increase the substrate concentration, you have extremely high, no effect you will see because enzyme to substrate ratio is very important. That you have studied in the biochemistry class. Please note that if you make an enzymatic process very efficient, there should be minimum enzyme and maximum substrate. Do you agree? If you want to load a lot of enzyme, the process efficiency is compromised because enzymes are also expensive. So you need to find a right balance between enzyme to substrate ratio. Here is one example showing the alkaline protease is an enzyme people have extracted. It has an optimal pH around 9 and optimal temperature around 60 degrees Celsius, just to show. You can do the enzyme kinetics yourself. All right, now coming to the real experiments here. This is the protein extraction done on rapeseed matrix, okay, with and without enzyme, okay. And this gray region is without enzyme and this is with the enzyme. You can see that the yield is significantly increasing when enzymes are added. They are trying to use four or five different proteases here. They have different activities here. You can clearly say that the enzyme will actually improve the yield. Microalgae, it could be rapeseed, it could be soybean. Enzyme acid extraction is actually a good extraction technique to use, improve the yield compared to ultrasound and pulsed electric field. Make sense? You will really see, I have done it several times myself, you really see the improved extraction yield when you use enzymes, especially with microalgae. They're rough and tough cell wall, enzymes can actually cleave them in a very efficient way. Okay. This is another example again with the rapeseed matrix. In this case, they use this is a no enzyme. This is with the enzyme called pectinex, contains pectinase. You can see there is a rapid increase in the yield. Okay. But usually people use the combined technique. That means you can combine ultrasound with enzymes. You do first ultrasound treatment, reduce the particle size. Do you agree? So, this is the big particle size, and you first do the ultrasound treatment, then the particle size is reduced. Then you add enzymes. What enzymes need to attract to the substrate. Reducing the particle size means increasing the surface area. There is a more enzyme to substrate interaction, and that will increase the process efficiency, otherwise extraction yield. Most often the recent article, they actually use combination of ultrasound and enzymes to improve the yield. There's a lot of studies that show that is a added benefit. Okay. So, you can use the combined techniques to use the improve the extraction yield. What are the advantages and disadvantages of enzyme assisted extraction? Yes, enzymes are specific. That's the beauty of enzyme. That's sometimes it's advantage, sometimes it's also disadvantage. Yeah.
Optimization is very necessary for the efficiency.
Exactly. For every single substrate, you need to optimize. You need to find out the optimal conditions. Yes, good. And he mentioned it is a green technology because enzymes are environmental friendly because they are produced from natural resources, no chemicals, and yeah. But enzymes are also expensive. It's not easy. And other advantage and disadvantage is that if enzymes can be used in liquid phase and also in immobilized phase. If it is immobilized enzyme, it's easy to remove them. If it is a liquid phase enzyme, they're always present with your protein plus enzyme. Maybe the enzyme is a problem for you. You never know what it will, if you eat enzymes at the end of the day, it might damage something in your stomach. We don't want to do that one, right? Because enzymes are continuously active. For example, if you add a cellulase, you continuously store it in room temperature for 10 days, you get glucose at the end of the day, and some people will take that in to do the spray drying, then you have all the Maillard reactions, the whole protein becomes brown. So, it's a problem. So, you have to inactivate them. To inactivate enzymes, again you have to heat them, that's a problem. You cannot add chemicals to inactivate enzyme because we are talking about food. Again, there is a problem. The post processing is actually a problem with the enzymes. How do we inactivate it? How do we remove the inactivated enzyme? Please note that in food industry, it's not a problem when the enzyme is inactivated. They don't care. Yeah, in food industries, it usually is not a problem if you inactivate enzyme because enzymes are proteins, but the active enzymes could be a problem. Okay, that's the reason why you should not add enzymes in a lot of quantity. All right, that brings to the end of my lecture. So, I'll try to conclude my today's lecture somehow. So, we started with what? Lecture 1? No, we started with marketing. Okay, market is increasing, keep that in mind. Then we started with the conventional extraction. We started about dry fractionation, good one, but it's also having its own problem. Salt extraction, salting in, salting out. Then we are talking about alkali extraction, high alkaline condition extraction, isoelectric point, precipitation, but in every cases we have to remove the excessive salt, it's a problem. And alkali extraction brings chemical changes, it might be a problem. Then we talked about non-conventional extraction, we covered ultrasound, cavitation phenomenon, very efficient technique to damage the cell, very effective to reduce the extraction duration. Then we went to for PEF, electroporation, making the hole, very efficient in some cases, but not necessarily in all the cases. Seeds, it's very difficult to damage them. Strong cell wall, you can't damage them. Then we talked about enzyme, very efficient because most of the enzymes are very efficient in cleaving the cell wall and removing the compounds out, but post processing is required. How do we inactivate the enzyme? How do we remove non-inactivated enzyme? That's a problem. That's it for today. Okay.
Comparison on the yield of these novel technologies.
Yeah,
means application of this novel technology.
There is a lot of papers. I haven't put that purposefully because I could not find which one is the best one to say. Everything showing that okay, there is an increment. This is better than the other one.
extraction technique has its pros and cons.
Exactly. Every single extraction has a pros and cons. Depending on what you want, what is the facility you have, what is the end goal of the work, use that technique. That's the overall idea. Okay? Okay, then we are actually having the tutorial session. I'll get back to you on that one later. Okay? That's the last session. The lecture is over for today.